This is the last part of the metabolism, and we'll discuss electron transport chain, especially their players, the mechanisms, how they transfer the electrons, and synthesize the ATP, which is the energy source for a living system. Now we'll deal with the process of electron transfer from reduced coenzymes such as NADH to molecular oxygen, and also we'll discuss how that can lead to ATP synthesis. This process is known as oxidative phosphorylation, which occurs in mitochondria, and it also requires oxygen molecule as well as functioning electron transport chain. The mechanism for the oxidative phosphorylation involves pumping the protons out of a mitochondrial matrix into the inner membrane space during the electron transfer. And this pumping out of the proton leads to a creation of an electrochemical gradient across this inner membrane, and that leads to the ATP synthesis. And basically this gradient is used as a power source for this ATP synthesis. And again, we're going to discuss more about the mechanism, how this ATP is synthesized, and how this system uses such a proton gradient for the ATP synthesis. Oxidative phosphorylation and the ATP synthesis happens in the organelle called the mitochondrion, or mitochondria as a plural form. They're basically an organelle, has the ellipsoidal structure, and they're sized 0.5 micrometer by the 1 micrometer or so, and there are roughly 2,000 fragments in the normal-sized animal cells, and they are oftentimes interconnected. When you look at the structure of a mitochondria, there are outer membrane, which is permeable for most of the things that are smaller than 10 kilodalton, and it contains porins, which works as a channel, so there are a lot of things that can freely diffuse in and out. And the composition is about 40% lipid and 60% protein. And the inside of the outer membrane of mitochondria, there's an inner membrane, which has the more complex composition. And when you look at their composition, there are 70 to 80% are the proteins, and they are mostly electron transport chain, or the ETC, or oxidative phosphorylation, or OP proteins, that are organized into multi-protein complexes. And this inner membrane is extremely folded to form this structure called cristae, and that provides the large surface area in a small volume, so it can accommodate a lot of important proteins, or the protein complexes, for electron transport chain. And this inner membrane is impermeable except for the oxygen, carbon dioxide, and water molecules, but it still has the transport systems for ions, substrates, coenzymes, and so on, which are controlled by the elaborate transport systems. And inside of this inner membrane of mitochondria, there is a matrix, basically a cytosolic area inside of the mitochondria. So it's a highly concentrated gel-like substance internal to this inner membrane. And its constituents are the water molecules, proteins, substrates, and cofactors, coenzymes, as well as different kinds of ions. And TCA cycle enzymes are located here too, except for the succinate dehydrogenase, which is located and bound by on the inner membrane of the mitochondria. So mitochondria, interestingly, has its own genetic system, meaning that it contains its own DNA, the mitochondrial DNA, but the mitochondria are not fully autonomous. So it needs the proteins and some enzymes, stuff like that, from the outside of the mitochondria that was synthesized by the actual nucleus. There are some issues in transporting of substrates in and out of the mitochondria. Like we discussed in previous slide, outer membrane has no problem because it's permeable to most of the things because it contains pore channels, so the diffusion works there. But the inner membrane is impermeable except for the oxygen, carbon dioxide, or water molecules, and also have a very high content of proteins, as 75 to 80% of the membrane consists of proteins. So it's pretty crowded with all those proteins, especially the electron transport chain proteins and transporters and so on. So there are not going to be any free diffusions happen. So the mitochondria need to somehow get the huge reducing equivalents such as NADH made in the glycolysis in the cytoplasm to the site of action for the TCA cycle, which is innermost side of the mitochondria in matrix. So in order for it to do so, it uses this system called the malate shuttle, where oxaloacetate in the cytosol is reduced to malate with the this NADH that is oxidized into NAD+, and then this malate is transported into the mitochondrial matrix using this malate shuttle, and then reoxidized into oxaloacetate for getting into the TCA cycle, and it also regenerates this NADH in the matrix. And also the mitochondria needs to get the ADP in to produce ATP, and also ATP that is already produced pushed out of the mitochondria. So that is done by the adenine nucleotide translocator, and it'll result in the export of the charge, one charge. So when ATP goes out, basically ATP has the 4 negative charges. So 4 minus, or the negative charge, goes out, and ADP comes in, meaning that only 3 negative charges coming in. So the net export is going to be the 1 negative charge per ATP that gets in. And this is driven by the electrical difference of the in and out of the mitochondria, especially the inner membrane or the matrix, due to the proton pumping. So basically the proton, the positive charge, goes out. And again, we're going to discuss about all these details and the mechanisms of them the later of this lecture. Function of electron transport chain in the mitochondria is based on the ability of coenzymes and other electron carriers to undergo reversible oxidation and reduction reactions, or redox reactions. And for those such redox reactions, electron donors and acceptors are required. And the direction of electron transfer would be from reduced partner. Here it says reducing agent, so they are referring the same thing. Reduced partner, which is reduced, so it already contains electrons, to the oxidized partners. Oxidized meaning that it already lost the electrons. So now it has ability to take those electrons to become reduced. So again, the direction of the electron transfer is from reduced partner, which is donor, to oxidized partner, to acceptor. And this reduced partner donor gets oxidized because now it loses electrons, while in a coupled process, the oxidized partner, which is an acceptor, gets reduced, which means it gains the electrons. And like I told you, it might be a little confusing, but this reduced partner is also called as reducing agent, because this is an agent that reduces other molecule, which is already oxidized here. And the oxidized partner, an acceptor, is also called as oxidizing agent, because it oxidizes the reduced partner, which is a donor. Let's revisit the thermodynamics to better understand how redox reactions work. The likelihood that electron transfer will take place and its direction are determined by a fundamental property of the reacting species called reduction potential, and it can be designated as this cursive E. and it actually indicates the tendency of a given reacting species to gain electrons while the redox partner gives them up. So it's about that tendency of getting reduced, because it's about getting the electrons. And negative E means that that particular species have a tendency to give up the electrons when paired with a certain standard. So this standard can work as a reference point for telling how much of this tendency of giving electron is. And typically the hydrogen reduction is used as a standard. And positive E means that that species has tendency to attract electrons when paired with the standard. And the standard reduction potential, written as E naught, is known for most things, which is listed in this table. Then how is this reduction potential E is measured? So it is measured by electrically connecting two reactants via a voltmeter, and among two reactants, the one will be used as a standard. So there's a sample half cell containing both reduced and oxidized forms of the reactant that are at 1 molar, so it's going to be standard condition, with a standard half cell, which is typically the hydrogen ion at the 1 molar and hydrogen gas at 1 atm. And these mixtures are also called as redox couples. So the voltmeter basically reports the direction and the magnitude of the electron flow, which reports basically the electromotive force. So if the electron moves towards the hydrogen atom and hydrogen gas half cell, that means this reactant tested are getting oxidized. So it'll have the negative E value, the reduction potential value. If that is the other way around, meaning that if the electron moves away from this standard half cell, that means the reactants gets reduced, and that means it will have the positive, that the reactants have a positive reduction potential, or E value. Let's think about the reduction potential relative to the standard reduction potential, which is indicated as E naught here. E naught is the electromotive force that is measured in volts at 25 degrees C, pH is 7, and the aqueous solution using a mixture of oxidized and reduced forms at 1 molar each against the standard half cell. And again, the standard half cell contains the hydrogen ion at 1 molar and hydrogen gas at 1 atm. And this coupled H plus H2 standard is assumed to have E naught of 0 voltage. Therefore, E naught is a measure of a potential or the tendency of a given chemical to be oxidized and reduced compared to the standard, which is the reduction of a hydrogen ion. When the E naught is positive, that means the reactant tends to be reduced. And for example, the oxygen has the highest E naught as 0.815 volt. And E naught is negative. If that's the case, that means reactant tends to be oxidized. But what actually really happens depends on the E naught value of the partner, not the standard. So the same substance can act as either donor or the acceptor, depending on the E naught of their partners for the reaction. The real redox reactions are coupled half reactions. So let's take a look at this reaction here, which happens in the TCA cycle, where the isocitrate is converted to alpha ketoglutarate through oxalosuccinate here. And the reaction actually is going to be the NAD plus and the isocitrate to be converted to NADH. So this is going to be a reduced form of NAD plus, and then hydrogen cation and alpha ketoglutarate and carbon dioxide. And this reaction can be written as two half reactions, but note that by convention, these half reactions are always written as reduction reactions, not oxidation reactions. So for NAD plus, it's going to be the same order as this reaction here, where NAD plus has 2 electrons and 2 protons to make the NADH and H plus, and NADH is again the reduced form of NAD plus. So this is a reduction reaction, as opposed to the other half reaction is written in the reverse direction. So alpha ketoglutarate has 2 electrons and 2 hydrogen cations and carbon dioxide to form the isocitrate again. Then this alpha ketoglutarate is reduced to isocitrate. And their ENOT values are here written here. And then now you have these two reactions, but which way will electrons flow? Is that going to be this forward direction or the reverse direction? In order for us to tell that, we should look at these ENOT values. In real redox reactions, electrons always spontaneously flow from the half reactions with more negative ENOT to the half reaction with less negative ENOT or more positive ENOT. So in this case, that's going to be the electron flowing from this half reaction to this half reaction. And the flow of the electron in the opposite direction is possible too, but it's going to be an endergonic process. Therefore, this isocitrate is a donor, and this second half reaction is actually moving in the reverse direction. So starting from the isocitrate, and then going to be oxidized to the alpha ketoglutarate. And we can define delta ENOT as the difference between the ENOT values of these two reactants. So ENOT of the acceptor minus ENOT of the donor. So if you plug those numbers that we got from the previous slide into this equation, then we're going to get 0.06 volt. And this parameter delta ENOT is called standard reduction potential difference. And the free energy change, which is a criterion for the spontaneity of the reaction for redox reactions, is given by equation here. Delta G naught equals minus NF delta G naught. Here, N is the number of electrons transferred in the redox reaction, and F, the cursive F, is the Faraday constant, which is 96,485 joule per volt and mole. And delta G naught is again the standard reduction potential difference, where you can get it by the difference of these ENOT values from the acceptor and the donor. Now note that since there's a minus in this equation, the positive delta ENOT value will result in negative delta G naught, and negative delta G naught is an indicative of a spontaneous reaction. So in the above example here, when we put the numbers for N, because there were 2 electrons that were transferred in half reactions, they're going to be number 2 getting into N here, and then you plug the Faraday constant as well as delta ENOT to get the minus 11.58 kilojoule per mole. So that's a negative value for delta G naught, meaning that this reaction is a spontaneous reaction. But there's a warning that some reactions in the cell proceed in the opposite direction relative to the one suggested by the consideration of this delta ENOT acceptor and the delta ENOT donor, because of the concentration differences. As we've discussed, if there's a lot of products that are gotten rid of from the other next reactions, then it will actually change that ratio, the KEQ values. So the reaction will happen in the opposite direction than you can get with just a simple this ENOT values. But in the conditions in the actual cells, in the real delta G naught of redox conditions, which occurs under conditions different from the standard conditions, 25 degrees C, pH 7, and so on, 1 molar concentration for the reactants and so on, is different definitely from the real situation. but this delta G naught in that case depends on the concentrations of reacting species. So to control those and take those into consideration, there is a new equation called the Nernst equation, where the delta E naught prime is calculated, and delta E naught prime is the real delta E naught in the cellular condition. So that is using delta E naught, but use other factors here, minus RT divided by NF multiplied by ln and ratio of the concentration of a product and concentration of a reactants. And in this case, then delta G naught prime will be the same here, minus NF multiplied by delta E naught, but prime, so which we got right here from the Nernst equation. So let's consider the reaction here, oxidized A and reduced B reacting to get the reduced A and oxidized B in the redox reaction, in which case the delta E naught prime will be delta E naught minus RT divided by 2, because here's our 2 electrons that are transferred from between reactants, so the N will be 2, and Faraday constant multiplied by ln, and again concentration of all the product divided by concentration of all the reactants, and the real delta G naught prime would be minus 2 Faraday, and the delta G naught prime value you got here. Then how is this all relevant to the electron transport chain processes that's happening in the matrix or the inner membrane of mitochondria? When we look at the NADH and FADH2, which are the coenzymes for such process, they transfer their electrons, working as a reducing equivalent and involved in redox reaction to ultimately hand these electrons to the electron acceptor, oxygen gas molecule, and energy obtained in this redox reaction, which is rather multi-step redox process happening in the electron transport chain, is used to drive ATP synthesis. So it's definitely involved in this energy generation for the cellular systems. And the ultimate reaction for such NADH redox reaction is NADH plus hydrogen cation plus half of the oxygen gas molecule is the converted to the NAD plus and water molecule. And if you divide this reaction into two half reactions, these are the two reduction reactions, one for the NAD plus and the other for the oxygen molecule, becomes to the water molecule here, and the E naught prime values are reported right here. And when you look at these E naught prime values, you can actually tell that how the reaction goes, if it's gonna be forward or reverse. So when you look at the delta G naught prime, again, this is the standard reduction potential difference. It's gonna be the E naught prime of the acceptor minus E naught prime of the donor. So we can plug in these numbers here to get the 1.13 volt here. And again, this value will get in here in this equation to get the delta G naught prime, which gives us minus 219 kilojoule per mole. So it's highly negative, meaning that this reaction is highly spontaneous. So this results in the release of a huge amount of energy here, and that is sufficient to synthesize about 7 molecules of ATP. When we consider that the synthesizing 1 molecule of ATP costs about 30.5 kilojoule per mole at standard conditions. But in the cellular conditions, the real example, when it's released in a smaller packets and happening in the actual cellular system, it's going to be about 3 ATP that is made per each NADH oxidized. This graph shows the how electron is moved in the electron transport chain. So started from this NADH, the electron is handed to the other components in the ETC electron transport chain to the oxygen molecule to get it oxidized to water molecule here. And again, it's always from the more negative E naught prime value to more positive E naught prime value that the electron is moved. And we're going to discuss more about this electron transport chains, their components, as well as how these electrons are moved through and generate the ATPs. So on the inner membrane of the mitochondria, there are overall two paths of this electron transfer. One is through the NADH, that electron moves to the complex 1, go through the coenzyme Q, and then complex 3, and then electron is transferred to cytochrome C and complex 4, and finally to the oxygen to get it oxidized to water molecule. And another path is the starting from the FADH2 in the complex 2, and then going through the pretty much the same path to the coenzyme Q, complex 3, cytochrome C, and complex 4 to the oxygen molecule. So let's take a closer look at these path of electrons in the mitochondria. And the first one on that path is complex 1, which is NADH coenzyme Q reductase, and also called as NADH dehydrogenase. And it basically transfers a pair of electrons from NADH to the coenzyme Q. And this coenzyme Q is also called as ubiquinon or UQ because it can be observed in almost all different kinds of types of cells. And the total reaction will be the NADH plus coenzyme Q, that is oxidized form, is going to be converted to NAD plus and coenzyme Q, that is reduced form, with the delta G naught prime value minus 69.5 kilojoule per mole, so it's spontaneous reaction, and delta E naught prime is here 0.36 volt. And this complex 1 is membrane-bound large multi-protein complex, which is composed of more than 30 polypeptides. And it contains FMN, which is flavin mononucleotides, as well as 6 to 7 iron sulfur clusters, and they can act as electron carriers because these ion can be either trivalent or divalent, so you can easily get another electron to become the divalent and recover back to the trivalent to give out the electron. And path of electrons will be starting from the NADH binds to on the matrix side, and then the complex 1 in the inner mitochondrial membrane will take the electrons from the NADH, so and that moves to the FMN to make the FMNH2, and then FMNH2 transfers through the series of these iron sulfur clusters. They're typically in the 2F2S form or 4F4S form, but going through the 6 to 7 of them to hand this pair of electron to coenzyme Q or ubiquinon. And coenzyme Q is a mobile carrier, so it diffuses in the membrane to complex 3, which is the next complex in the path of electron transfer. Complex 1 is peculiar because there is a switch from 2 electrons that was handed from the NADH to a single electron transferred mediated by FMN. So the actual reaction here for the NADH oxidation will be NADH plus H plus is NAD plus 2 hydrogen cation and 2 electron. Again, here NADH, the H is gonna be the hydride ion, which is a negative charge, the anion of the hydrogen. So there are gonna be 2 electrons that is contained within this coenzyme. So that's why you're getting 2 electrons. So NADH is exclusively 2 electron carrier, but the iron ion in this complex 1 is exclusively 1 electron carrier. So there's a discrepancy between these two, make it difficult to hand these 2 electrons directly to the iron sulfur cluster. But the FMN can either be a 2 electron or the 1 electron carrier. So it serves as a link between these two, NADH and the sulfur iron sulfur cluster. And complex 1 provides a sufficient delta G naught to catalyze production of ATP molecule, and also the extra energy are going to be released as heat. But making this ATP molecule by complex 1 happens indirectly, because this complex 1 itself does not have ATP synthase or so, but it sets up the proton gradient across the inner membrane by pumping out the protons out of this matrix, and this proton gradient will be later used for synthesizing ATP within this electron transport chain. When you look at the stoichiometry of the number of protons that are pumped out, there are 4 hydrogen cation that is transported from the matrix to the intermembrane space, and they are called as N side and P side respectively. And this 4 proton is transferred per each 2 electron passed from the NADH to eventually to the coenzyme Q. And there are inhibitors that are directly only specifically affecting this complex 1. One example is rotenone, which is a plant toxin used by Amazon tribes to poison fish, and the other is amytol, or the barbiturates. Electrons can also be introduced into electron transport chain from the FADH2 rather than NADH2 using complex 2. And it's also called as a succinate coenzyme Q oxidoreductase, which contains a succinate dehydrogenase, and that's the only enzyme of TCA cycle that is membrane bound. So the reaction is right here for the FADH2 gets into the complex 2 to provide the electrons. So FADH2 plus coenzyme Q oxidized form converted to the FAD and the coenzyme Q reduced form with the delta G naught prime value as minus 2.9 kJ per mole, and delta E naught prime is right here. And this complex 2 is a large protein complex of 4 subunits, and it contains a covalently bound FAD within that complex, and has the 3 iron sulfur clusters. And path of electrons will be from the FADH2, and then electron moves to the iron sulfur clusters, go through these, and then coenzyme Q, and later on this electron will be moved to the complex 3, which is not shown here. But it does not, this complex 2 and the FADH2 path does not release enough free energy to synthesize ATP. So, and also there's no proton pumping occurs here. So there are not going to be any proton gradient set up by this complex 2. But it only serves as for injecting the electrons into the electron transport chain. So the next player in the path of electron transfer is coenzyme Q, also known as ubiquinone or UQ or UQH2. This is a mobile electron carrier, and it accepts either 1 or 2 electrons, and it has a hydrophobic tail, so it can diffuse freely right here. So it can diffuse freely in the inner membrane. It actually accepts the electron from this complex 1, which is shown right here. And it serves to shuttle the electrons from complex 1 to through the this coenzyme Q to complex 3, or the complex 2 to the complex 3 here. The next one is complex 3, which is also called as coenzyme Q cytochrome C reductase, and basically it transferred the electrons from coenzyme Q to cytochrome C. Here's the reaction here with the delta G naught value as minus 37 kilojoule per mole, and the delta E naught prime values right here. And it's part of, again, the inner membrane of the mitochondria, and it contains two type B cytochromes, which are BL and BH, and H is for the high, and the L is for the low of the E naught value, and also it contains the cytochrome C1 and one iron sulfur cluster. So the path of electron transfer is complex, and it's called as a Q cycle, and it provides sufficient free energy to synthesize ATP molecules, and it also acts as an unbalanced proton pump. So for each 2 hydrogen cation taken on the matrix side, 4 hydrogen cation is released on the cytosolic side. So there are going to be difference here as of the 2 protons, but this balance is corrected by the complex 4, which comes the next on this electron transfer chain, which also is the unbalanced proton pumping, but in the opposite way. So at the end, there's going to be a total balance of the proton number of proton that is pumped there. There's an inhibitor that is specifically working on the complex 3. An example is an antimycin A, which is an antibiotics. And the next one up after the complex 3 in terms of electron transfer is cytochrome C, and this is the only electron transfer chain component that is water soluble. It is a small globular protein and loosely associated with the inner membrane, and it has a porphyrin ring and serves as a mobile electron carrier. So it moves along the surface of the membrane and then basically gets the electron from the complex 3 and then deliver it to the complex 4, and it's exclusively one electron carrier. And the last component in the electron transport chain is complex 4, and it's also called as a cytochrome C oxidase, and it accepts electrons from cytochrome C and delivers that to the oxygen molecule. So the reaction is basically handing the 4th electron to the oxygen molecule to get it reduced to water molecule. So here's the actual reaction with the delta G naught value as minus 110 kilojoule per mole, so it's highly spontaneous too. And here's the E naught prime value, and it also spans the inner membrane. And this complex 4 is composed of 10 or more subunits, including cytochrome A, cytochrome A3, and the 2 copper atoms, cycling copper mono and divalent. And it's on exclusively one electron carriers, and the path of electron transfer is starting from the cytochrome C. Electron moves to the copper, which is here, the copper A, and then cytochrome A, and then moves to the copper B and cytochrome A3, and finally goes to the oxygen gas molecule here, the half of the gas molecule here, to make it into the water molecule. And it generates the enough energy to synthesize ATP molecule. And this also, like we talked in the previous slide, acts as an unbalanced proton pump. So it takes 4 proton on the matrix side and releases 2 proton on the cytosolic side per 2 electrons that is transported, basically from the complex 1. And inhibitors that are working specifically on the complex 4 are cyanide, azide, and infamous carbon monoxide. Then how is the ATP synthesis powered? When you look at this electron transport chain, this is definitely exergonic with a lot of negative values for delta G naught prime. But how actually is this energy is used for ATP synthesis? So there has been search for the high energy intermediates which can be used for their hydrolysis to make the ATP synthesis, drive the ATP synthesis. But after those searches, they came up with nothing. So there wasn't any high energy intermediates that obviously located there and involved in the ATP synthesis. So to explain how this system makes the ATP, Peter Mitchell in 1961 proposed this chemoosmotic hypothesis, and he earned the Nobel Prize in the 1978. And this chemoosmotic hypothesis basically proposes the view that the energy of electron transport is conserved by creating an electrochemical proton gradient, especially between the matrix and the inner membrane here, so inner membrane space. And that electrochemical gradient drives the ATP synthesis outside of this electron transport chain, but somewhere else on that region. Then where does the energy come from? We can calculate the delta G for transporting one molecule of protons from the matrix to the intermembrane space by using this equation. Well, delta G equals 2.3 RT multiplied by the difference of a pH inside and outside of the inner membrane. And remember that there's a 2.3 factor coefficient that is multiplied here because there was basically the conversion from the ln, the natural log, to the pH, which is log of 10. So there's a ratio of 2.3 differences there. So that's why you get this number here. And also there's another factor that is added here, which is a Z multiplied by F and the delta psi. Here Z is the charge of a proton. So in this case, that's going to be 1 because it's one molecule of a proton. And F is a Faraday constant. And delta psi is the electrical potential across the membrane, and it's positive if protons are pumped out. So pH is typically lower outside than inside of the inner mitochondrial membrane. And let's plug in some typical values here, where delta pH of the inside and outside of this inner mitochondrial membrane is 1 unit, and delta psi is 0.18 volt. Then at 37 degrees C, which is our body temperature, we're going to get the delta G is 23.3 kilojoule per mole. So that is to say that the proton transfer from the matrix to inner membrane space is an endergonic process. So that is the direction of the proton moving from inside the matrix to the outside, right? So the reverse transport or the movement, basically the proton that is discharged from the inner membrane space, so it moves from the inner membrane space to the matrix, would be the exergonic process, which is the opposite of what we got here as 23.3. So that's going to be delta G minus 23.3 for that direction, again from inner membrane space to the matrix. So that will provide an energy that drives the ATP synthesis. ATP synthase is the system that is capable of using such energy to convert ADP to ATP, and ATP synthesis costs 30.5 kilojoule per mole in standard conditions, but if concentrations are accounted for in the actual cellular conditions, the cost will go up to about 40 to 50 kilojoule per mole. So that means between 2 and 3 protons must be transported to release sufficient energy. Again, one proton can give minus 23.3 kilojoule per mole. So if there are 2 or 3 protons, that's going to be twice or 3 times of that, and that should cover this minus 40 to 50 kilojoule per mole, which is required for synthesizing one molecule of ATP. And chemoosmotic theory is consistent with a number of experimental observations. So what they've done is to isolate the intact mitochondria, but in this case, those mitochondria must have the intact inner membrane. Otherwise, all the ions can diffuse in and out to dissipate the electrochemical gradient. And also, inner membrane should be impermeable to ions such as hydrogen cation and potassium ions and so on, because their free diffusion would also dissipate the proton gradient. So in the isolated intact mitochondria, they've measured how the electron is transported, how much of the proton is pumped out, and found that did the measurement, and indeed found that there's such electrochemical gradient set up there, and also the numbers that they measured is quite consistent as the number that we got from this theoretical values. And also they've treated these mitochondria with compounds that increases ion permeability across the inner membrane. So that actually still allowed the electron transport, but the oxidative phosphorylation was inhibited, meaning they can again give the clues on the mechanisms of this, how ATP synthase works and the electron transport chain works for generating the ATP. ATP synthase is also called as proton translocating ATP synthase, proton pumping ATPase, or F1, F0 ATPase, because it has two portions here. The one is F0, which is water-soluble transmembrane protein that is bound on the inner mitochondrial membrane, and it has three kinds of subunits, and its stoichiometry is A1, B2, and C9 to 12. And it forms a transmembrane channel through which the hydrogen cation flow to F1, the other big chunk of this ATP synthase protein. And it's basically the proton is moved down the electrochemical gradient from the inner membrane space to the matrix of the mitochondria. And F1 is another huge part of this ATP synthase, which is water-soluble peripheral membrane protein, and it has the ATP synthesizing activity, and that this F1 is sufficient by itself for making the ATP. So especially this beta subunits make the ATP, but also alpha subunit binds the ATP, and its composition is 3 alpha, 3 beta subunits, as well as gamma, delta, and epsilon subunits. And its catalytic site is this alpha beta subunits, but the delta and epsilon controls the interactions of F1 with F0 within this ATP synthase complex. And also the gamma forms a stalk, serves as a gate controlling. And again, the proton, the hydrogen cation flows from the inner membrane space to the matrix through this ATP synthase, going through the F0 to the F1. So let's talk about the binding change mechanism for the ATP synthase to generate the ATP. So when you focus on the F1 complex, and there's 3 alpha beta protomers, and they each have the distinct sites for binding the ligand, especially ATP and inorganic phosphate. And also they are in 3 different conformations or the state. And those conformations are O state, which is an open, basically open state, and it's an inactive in terms of synthesizing ATP. Also, it cannot bind ligands, especially ATP and inorganic phosphate. Another state is the L state, where it has the loose binding of the ATP and inorganic phosphate, but it's still inactive form. But the T state is now having the tight binding of the ATP, and it's an active form. So energy in the form of a protein gradient is required for ATP release, but not for the ATP synthesis per se. But the without energy input here, there are not going to be any ATP that is generated. So it consumes the energy, but also it requires that proton gradient set up between the matrix and the inner membrane space, in order for the ATP to release ATP synthase release the ATP. Here's the sequence of steps for the ATP synthesis. So for the step 1, there's a ADP plus inorganic phosphate that binds to the L site, which is a loosely binding site, and previously synthesized the ATP sits in the T site, tightly binding site, and O site is empty. And in step 2, there's an energy input there, which drives the simultaneous conformational change of all these 3 protoforms. So polymers. So the L form changes to T form, and T form changes to O form, and O form changes to L form. So they're all of their state changes at the same time. And upon the L to T changes, the ATP is synthesized in T, and upon the T to O changes, ATP is released from O, and upon the O to L changes, which was empty, now it becomes ready to bind the ADP and the inorganic phosphate to make it into the ATP. So basically, after 2 more cycles, the enzyme will return to its original state. And there are several ATP synthase inhibitors, such as oligomycin B and DCCD, and both binds to the subunit of the F0, which is not shown right here, and it inhibits the proton flow from intermembrane space back into matrix and prevent ATPase activity. Once ATP is made by the ATP synthase in the mitochondria, it must be exported, and more ADP must be brought into the matrix to generate more ATP. But charged molecules such as ADP and ATP cannot cross the membrane easily by themselves, because the membranes are basically nonpolar lipid bilayers. So there's a system required to move those and transfer those ATP and ADP molecules. So ADP-ATP translocase does that, and it has one nucleotide binding site, so it can either bind ATP or ADP at a time, and it alternatively faces a cytosolic side or the matrix side. So the sequence of steps will be this ATP-ADP translocase binds ATP in, and then flips, and then release ATP out, and then binds ADP outside of the membrane, and then flips over, and releases ADP into the matrix, and then this cycle keeps circling going. And ATP has 4 negative charges, and the ADP has 3 negative charges when you look at the molecules, their structures. One cycle is equivalent to the exporting 1 negative charge, because out of -4 goes out and -3 gets in. So it's same as the importing 1 positive charge into the matrix, and this charge is transported down the electrochemical gradient, so it's a favorable process. We can use PO ratio, which is phosphate to oxygen ratio, to estimate the number of ATP molecules that are made per 2 electron that is transported through the electron transport chain. So the PO ratio is the ratio of ATP synthesized to the oxygen reduced. And you can think of it as analogous to PE ratio, which is price earning ratio. So putting the input and output ratio basically. And one molecule, ATP molecule synthesis requires movement of 2 to 3 protons into matrix from the energy considerations that we already discussed earlier this lecture. In addition, translocation of one ATP out and one ADP into the matrix is equivalent to the moving one proton into the matrix, which we also discussed in a few slides back. So 2 to 3 protons plus 1 proton equals 3 to 4 protons, and you're getting 1 ATP, right? So movement of like up to 4 protons down the gradient is required to make 1 ATP. So now movement of 2 electron, which is equivalent to the final product, which is half of the oxygen gas molecule through the electron transport chain, result in pumping out of a total 10 protons. So 4 hydrogen cation from the complex 1, 4 from the complex 3, and 2 from the complex 4. In total, it's going to be 10 H+, right? So the PO ratio would be 10 proton divided by 4 proton, which is required here to get the 1 ATP, equals 2.5 ATP molecules per 2 electron transported for this ETC, or also it's equivalent to the 1 oxygen atom, which is the half of the oxygen gas molecule. If started from the complex 2 right here through the FADH2, since this complex 2 doesn't give out or transfer any protons here, that's going to be 6 in total, 6 H+ in total. So the PO ratio is going to be 6 divided by 4, which is 1.5 ATP molecules per 2 electrons. Uncouplers are proteins or the compounds that uncouples the electron transfer process and the ATP synthesis. So these uncouplers basically inhibit oxidative phosphorylation as a result, and they act by dissipating the proton gradient across the inner membrane. Thus, uncouple electron transport from oxidative phosphorylation. And in here, the electron transport still occurs. So there are the electron that goes through the electron transport chain, but the oxidative phosphorylation, which is the ATP synthesis, is going to be blocked. And they do not bind to any of the proteins that are relevant to electron transport chain or the ATP synthesis or ATP synthases. And examples of such uncouplers are 2,4-dinitrophenol, dicumarol, and FCCP, and all they are hydrophobic and have dissociable protons, and this is important characters that they have. So they carry these protons from outside into the matrix of mitochondria and thus dissipate the gradient. So there are not going to be any proton gradient buildup because both sides will contain the protons, and both sides are going to be the inside and outside of the inner membrane spaces and the matrix of the mitochondria. An electron transport chain continues to function and results in proton pumping out of the matrix, but the protons immediately leak back into matrix, which results in the dissipation of the proton gradient. So there are not going to be any ATP synthesis occurs again, which depends on the relies on the proton gradient. So a lot of energy that was basically generated and stacked from the electron transport chain will be released as heat. And there are endogenous uncouplers that are basically encoded by the genes, and they are used by newborn mammals and hibernating animals to keep them warm. And an example is a protein thermogenin in mitochondria of brown adipose tissues. And they're brown fat cells because they have that particular brown pigmentation due to a large number of mitochondria content per cell. And they basically uncouplers create the passive proton channels in the inner mitochondrial membrane, which again dissipates the gradient of the proton. So a lot of energy generated from the electron transport chain will convert it to the heat and released from the cells. This oxidative metabolism is nice in a way that you can generate a lot of energy and store that as in the form of ATP, but also it comes at a price, which makes the free radicals. So cells need to come up a way to deal with them. So the free radicals are the chemical species with unpaired electrons in the outer shells. So that means they are the chemicals that have a lot of need or the tendency to have one electron from the other molecules around it for completing its octet structure. So since it has the tendency to take up the electron from the other species, it tends to oxidize various things to initiate chain reactions. And classical example for the free radical reaction from the organic chemistry is the halogenation of methane by chlorine. So it happens readily in presence of sunlight. So here's an example of the typical free radical chain reaction. And here in the also next slide, the asterisk is used to represent an unpaired electron of the free radicals. So here there's a chlorine gas molecule, and it happened to be turned to the two chlorine atoms that are radical now. So they need just one electron to complete the octets. And this two chlorine, one of this chlorine atom that is radical, the free radical, can react with this methane to oxidize it and make it into the methyl radical, and also produce this hydrogen chloride. So it can be acidic in that condition, and also this methyl radical can now react with this chlorine gas molecule to form the chloromethane, and also leave this free radical of chlorine again, and so forth. It will generate more the free radicals. Here's another example. So what happens if incompletely reduced oxygen escapes from cytochrome C oxidase somehow? Then you're going to get things like a superoxide radical here that is basically from this oxygen molecule or the oxygen gas molecule that obtains the additional electron here. So this superoxide radical is extremely strong oxidant and can react with many other things. And it also forms an even stronger oxidant, which is a hydroxyl radical in the reaction with the hydrogen peroxide. So you can see the reaction right here. And these free radicals readily remove electrons from various molecules, and that can lead to oxidative damages, which is detrimental to the cells. So when the oxidative damages are on the lipids, it'll lead to disruption of the membranes. If there's an oxidative damage to DNA, then there could be some point mutations. An example would be guanine changes to 8-oxoguanine. In this case, instead of G pairing with C, it'll pair with adenine. So it'll immediately make the introduced in this point mutation, and that'll be inherited to the next generation or the next cell division. And also there could be the oxidative damage to the proteins, in which case these proteins or the enzymes are going to be non-functional and or malfunctional. And there are antioxidants that basically remove such free radicals. So the first line of defense for such removing the free radical is a superoxide dismutase or SOD. and it catalyzes the reaction here where the superoxide is going to be converted to the hydrogen peroxide and the oxygen molecules. And SOD is very efficient because its ket Km, the ratio, is 2 times 10 to the 9 molar and the second reciprocal. So it's close to the diffusion controlled limit. So which means that as soon as this SOD meets its substrate, which is the superoxide, it'll immediately turn it into this hydrogen peroxide. And motor neuron degeneration in Lou Gehrig disease, which is also the ALS, can be caused by a dominant mutation in SOD gene, which acts as a peroxidase. So the mutation in this SOD genes will lead to a lipid oxidation because it cannot get rid of this excessive superoxide. So that eventually leads to the cell death of the neurons and the causes this Lou Gehrig disease. So the product of SOD reaction, which is hydrogen peroxide, is also potentially dangerous because it can yield reactive oxygen species too. So this hydrogen peroxide can be also removed by the catalase, which catalyzes this reaction to get the water molecule out of hydrogen peroxide, and also can be removed by the glutathione peroxidase for this reaction. So this picture shows the highly simplified representation of a metabolism where it has two big parts, the catabolism and anabolism. And catabolism is about the degradation or breaking down the complex metabolites into simple products. And through that process, those metabolites are oxidized by the help of coenzymes such as NADP plus or NAD plus. And they're gonna get reduced by themselves, and then there are gonna be a lot of energy that are generated during this process that are gonna be stored in the form of ATP. And the other half of the metabolism is the anabolism here, which is a biosynthesis from the simple products to build it into a more complex substance or the metabolites. In this case, the cells are going to use the energy from the ATP, so ATP converts back to the ATP in this case, and it requires electrons to get reduced of these simple products, and NADP or the NAD, those kind of coenzymes provide electrons that are needed for the reduction. So hopefully you have the sort of good big picture of how metabolism works in the cellular system, and especially understand how catabolism works. And for such catabolism, and especially metabolism of the sugars, we talked about this glycolysis and the citric acid cycle, and also oxidative phosphorylation all generates the energy in either ATP form or the form of the coenzymes that are going to be further used for the oxidative phosphorylation to generate much more ATP. And also the proteins as well as the lipids can be fed into this citric acid cycle to generate the more energies for the cellular systems. So hope you can get all the details and understand how this system works, and also can understand and memorize what the enzymes and the metabolites are involved in these processes.